time. So this is our virtual MDA Engage Community Education Seminar, where we're highlighting specialists in the Upper Midwest today. And I am Nicole Petrowski. I am MDA's Community Education Specialist. So we definitely appreciate you joining us today. We're committed to community education, and we believe that having these opportunities to learn from our specialists and having the opportunities to connect with others is very important. Our community education series, which is what we're having today, is geared towards general neuromuscular disease topics. And while each event features regional specialists covering these topics, we also have a specialty topic for each one that you can see on the screen. And we only have 2 more left for the rest of the year, and our SMA Symposium is going to be on November 14th. And then some of you might remember our MDA Telethon with Jerry Lewis. Well, we are excited to relaunch the legendary Jerry Lewis MDA Telethon with Kevin Hart joining as the host. The telethon will be a 2-hour special airing globally tonight at 8 p.m. Eastern, streaming live on the LOL network on YouTube. And funds received from this telethon will support the work that MDA does for the 43 neuromuscular diseases that we support. And we would not be able to put on this educational programming without the support of our generous sponsors. So I would like to thank NewMotion Foundation, Biogen, and Genentech for their support. And this is the schedule that we have for this morning. I do want to do just a few housekeeping items. We are recording today's event, and we will be posting it to mda.org's website for on-demand viewing in a few weeks. And we will be having a Q&A session at the end of each presentation. So please utilize the Q&A icon on your screen to type in any of your questions. And we also encourage participants to communicate with each other by utilizing the chat feature throughout the morning. So if you'd like to do that, please feel free to. And you will notice on the agenda there are a few little breaks between some speakers that will give me time to get the next speaker's audio and camera check. So you might hear us talking in between sessions. And then finally, I will be sending out a brief survey afterwards. So I would appreciate any feedback that you could provide on what you heard today. We want to make sure that we're discussing topics that are of interest to our audience and are relevant. And so your feedback helps us improve for programming for 2021. And with that, I am going to introduce Dr. James Dowling. He is a clinician scientist who is focused on gene discovery and therapy development. His research has examined questions of disease pathogenesis and therapy development for congenital myopathies and childhood muscular dystrophies. And his laboratory has helped pioneer the use of the Zebrafish model for these disorders. So, Dr. Dowling, I will turn it over to you.
Thank you very much for the invitation. I'm very excited to speak to everyone today. My name is Jim Dowling. I am a clinician scientist in Toronto at the Hospital for Sick Children. I'm very familiar with Midwest neuromuscular care, as I was an MDA clinic director at University of Michigan for 8 years. So I'm quite familiar with the landscape, and I'm very happy to be talking to everyone. My research group does work across several different model systems and on several different disorders, but I thought it would be interesting today to focus on zebrafish because it might be something that many of you have not heard much about. So I first want to present my view of drug development or therapy development for childhood muscle disease. This gives me an opportunity to highlight 2 things. First is to acknowledge and thank all of the patients and families that have contributed to our work, and also to drug development and neuromuscular disease in general. Obviously, the motivation for all of the work that we're doing is to help find therapies for your children and for you. We appreciate all the hard work that goes into that on your end as well. And also gives me an opportunity to thank MDA, both for inviting me, but also for sponsoring my research throughout the years. My very first independent grant was from Muscular Dystrophy Association, and so I'm very thankful for all of the support that they provide for basic scientists. So when I think about therapy development, I really think about it in 2 things in parallel, and today I'm only going to focus on the left half of the screen, which is the target development, and that's the process of finding a drug to bring to clinical trial and hopefully to patients for clinical treatment. But it's important to recognize that in parallel and equally as critical is the idea of developing understanding of disease and clinical trial readiness. And this is something that I think we as a field are really coming to appreciate more and more recently about the importance of that. And I'll highlight today where model organisms such as zebrafish fit into this process of the target development side, both from the characterization of disease, also impacting on the very first step of the clinical trial readiness part, which is really establishing the genetic basis of disease. And then talk about some role for fish in pathomechanism discovery, as well as probably the most important aspect of our use of it, which is for the identification of potential therapeutics. Okay, so just want to introduce. Have been greatly aided by the I'm sorry. I have a weird. Okay, I just wanted to introduce some of the model systems that we use in our lab, and these are certainly not the only model systems that are available, but I think it gives the appreciation of what the breadth of systems are that exists. I think many are familiar with the mouse as a model system. For a lot of drug development in neuromuscular disease, it is the go-to system, and that's because of the genetic similarity to humans, the similarity in terms of muscle structure and function, and the ability to genetically manipulate mice, which compared to other mammals is really a unique ability. There are also smaller critters which have important roles. So we recently did a study using a worm model of RYR1 related myopathy. And there are some advantages to this invertebrate, though it is quite genetically distant to humans. And then kind of an intermediate between that is the zebrafish, which I'll talk in much more depth about. And of course, there are other models. So we work quite a bit on patient-derived cell material, which provides some great advantages related to similarity to the human disease, as well as ease of manipulation. Once the cells are grown, they can be used for quite a number of different things. And probably also people are familiar with some of the larger animal models that have been instrumental in drug development. And in particular, for example, some of the dog models of, for example, Duchenne muscular dystrophy or X-linked myotubular myopathy, who have played pivotal roles in the development of some of the new therapeutics. So you might ask yourself a question, why would one use zebrafish? What can this little guy, and I'm going to show you a video of him swimming away, tell us that could help understand how we could potentially understand disease or treat patients such as these? And these are examples of 4 patients from my clinic in Michigan. So hopefully now I'll convince you as to why the zebrafish could be a model that one might want to use to study human neuromuscular disease. So firstly, and this might be a surprise, zebrafish are much more similar to us than you might think. So if you look at the genetic level, 70% of all human genes are found in the zebrafish genome. And if you look just at ones known to be associated with neuromuscular disease, it's closer to 90%. In terms of the biological similarity, zebrafish are a vertebrate, so they share that in common with us, and have many of the same organs that we do, including a chambered heart, striated muscle, liver, kidney, pancreas, and brain. They don't have quite similar, their lungs are different than ours, so it's not a great model for pulmonary disease. And how that may factor into understanding neuromuscular disease is something we haven't really explored too much. So from a genetic perspective, they turn out to be a very good model for studying muscle disease. One of their prime advantages has to do with numbers. So when you think about a typical human pedigree, you think about, most families having 1 or 2 children. Some will have more, but usually not more than, say, 3 or 4. Whereas in a zebrafish, when we have a mom and a dad we put together, they can have anywhere from 100 to 300 offspring. And those offspring, and they can have new babies, new embryos, about once a week. So you can imagine the power of study that we can do if we can generate in 1 month, say, 1,000 offspring, and how we can use that to the advantage for rapid study of disease and therapy development. And not only do they have many offspring and can do them frequently, but zebrafish also develop very quickly. So one can study muscle and nerve-related phenotypes very early in the development. So pretty much all of muscle development is completed in the first week of life. And most zebrafish with muscle disease phenotypes actually will not survive beyond the first 2 weeks of life. So this gives a window of opportunity to study that's compressed, but also sufficient for drug development studies, but also very rapid. So you can imagine that if I can generate 1,000 embryos every month and I can study mature muscle in them within a week, that creates quite an amazing power to do investigations. And this is just a video showing, oh, actually, so it's not going to show. If you could see this video, it's going to show how the zebrafish develop very quickly, and you can get muscle contractions within 24 hours, or sorry, 48 hours of the fish being born. Equally as important, and as similar to mouse, zebrafish is a very genetically tractable system. And what I mean by tractable is that we can introduce mutations, we can introduce fluorescent tags, we can manipulate the genes that are potentially associated with neuromuscular disease to create models that genetically are identical to human disease. So I've shown as an example on the right a recent study that we did where we made models of several different mutations in the Dynamin-2 gene, which is associated both with centronuclear myopathy and with Charcot-Marie-Tooth disease. And we made each of the individual mutations in the zebrafish and then studied their effects. And we're able to show, for example, that the mutations associated with centronuclear myopathy caused one set of changes that was different than the ones associated with Charcot-Marie-Tooth disease, which is what you might predict based on the different human phenotypes. But we were able to do this in a rapid fashion and to accurately model the human genetic situation. Importantly, human or zebrafish is very good for studying muscle. And that's because the muscle structure and function in the zebrafish is basically identical to that of human muscle. So all of the known structures are present: the sarcomere, the triad, the Z-band, the dystrophin-associated glycoprotein complex, and all of the known muscle disease proteins are present and seem to function in the same way in zebrafish muscle as they do in human muscle. Also, we've been able to show that several of the very classical pathological changes seen in human muscle disease can be recapitulated in zebrafish muscle. So in the bottom panel here is a zebrafish nemaline body that is basically looks identical at the level of the electron microscope to what you would see in a human nemaline myopathy muscle biopsy. So we can recapitulate all of these pathological features, suggesting that the same disease mechanisms are preserved in zebrafish as they are in human muscle. As a caveat, I should mention that aspects of the nerve input to muscle are different in zebrafish than they are in human. And the biggest difference really is the fact that the peripheral myelination is composed of different proteins than in humans. So for example, one of the most common causes of, actually the most common cause of CMT in humans is duplication of the PMP22 gene. But PMP22, while found in zebrafish, is not expressed in the peripheral nerves, but rather in the nerves in the brain. And so myelination is different. And so it turns out to not necessarily be a great model for Charcot-Marie-Tooth disease in particular. Just to show that not only is the structure the same, but we can visualize some of the very dynamic processes. This is looking at calcium imaging inside the zebrafish muscle in a live fish, and showing, first of all, on the top panel here, that we can do these very interesting dynamic recordings in the live muscle, which is something that's basically impossible to do in other model systems. So you can't look at live muscle in the mouse in the setting of the intact animal. This is just to show that we can also see abnormalities. So this is, again, our model of Dynamin-2, where we know this intracellular calcium release is impaired, and we can recapitulate that in the zebrafish. And finally, it's a good model for looking at some, I'd say, cool features of muscle disease. So one one phenotypic change that we know happens in some congenital myopathies is paralysis of the eye muscles in a feature called ophthalmoparesis. And this is something that you can't really see in mice because mice don't move their eyes in this way. So we were able to see this in zebrafish, and I'll just show you real quickly. This is a wild type, a normal zebrafish, and you can see it was following an optokinetic drum, and so hopefully you can see the eyes shifting back and forth. The eyes are these big things here. And this is a model of X-linked myotubular myopathy, where in patients we know that there's eye muscle paralysis, and you can see here the fish, the eye muscles don't move in response, so it has complete eye muscle paralysis similar to patients. To us, our only logical conclusion is that zebrafish are a great model for studying human neuromuscular disease. We've used them in particular in 3 areas. One is to study disease pathomechanisms, and by which I mean the things that happen in muscle that are not normal in the disease setting in comparison to normal or wild type muscle. And that gives us an opportunity to use those pathways to understand the disease, but also as a springboard for therapy. And I'll show you an example of that. Another is for gene discovery and gene validation. So as you're probably aware, we're continuing to discover new genetic causes of neuromuscular disease. And sometimes when one discovers this, it can be unclear whether or not the genetic change is truly a cause of disease, or rather just a rare variation in an individual that's not associated with disease. And fish have proven to be a very powerful model for providing validation of those new gene mutations. And finally, and the focus for today will be about drug discovery. So I just should mention that we've developed a whole phenotyping toolbox for zebrafish models of muscle disease, and this is something that we've widely published on and shared with many other groups, and other groups have also helped in this development. So I think as a field, we're getting to a point where there's a lot of standards across different labs that are being used as a means of analyzing new zebrafish mutants. So I just want to give a few examples of how we've used this before I talk specifically about the drug discovery. So just an example of how zebrafish have been used to validate a new genetic cause. This is from a study now back in 2014 led by Misha Yuen and Nigel Clark in Australia. And they identified the first patients with nemaline myopathy and mutations in this gene called LMOD3. These are actually the first 2 individuals that they found with these mutations. And as part of an effort to prove that these mutations were the actual cause of disease, we made a model of LMOD3 in the zebrafish and showed that it caused disorganization of the sarcomere and the presence of nemaline bodies and nemaline rods. So providing good validation observation of the pathologic nature of LMOD3 mutations. And now it is an accepted cause of nemaline myopathy in patients. To talk about the pathomechanism part, I want to highlight that zebrafish can provide some opportunity for discovery of new observations of disease that we didn't know about before. Though this is a relatively rare occurrence, I think it is still an important thing to note and does emphasize how the fish is an important model. So one example, and this came from our group, where we found upon making a zebrafish model of X-linked myotubular myopathy that a specific structure called the triad was abnormal. That's shown here on the electron micrographs on the left. Here's a normal triad. And then here are 3 examples in 3 fish of abnormal triads. Well, it turns out that this observation had never been made before, despite the fact that the disease had been known for many years, and many people had looked at the electron micrographs of patients and just never seen this observation before. We went and looked at several human muscle biopsies, and lo and behold, found the same changes there, thus showing that this was a conserved abnormality. This was in 2009, and if you fast forward to now, these triad abnormalities are considered the most important abnormality in the muscle of X-linked myotubular myopathy, and are really the target of all therapies, is trying to improve the triad in this disease. And not only in X-linked MTM, but also the other forms of central nuclear myopathy, triad abnormalities are the key abnormality. And this all came first from an observation in zebrafish. All right, so I'm going to spend the last part of my time talking about therapy development using the zebrafish. And so you can think about this in really 2 general ways. One is a targeted approach, where you have a specific either pathway, genetic target, something where you have some advanced knowledge and you're going to say, well, I want to do a specific drug that goes after that advanced knowledge to see if it impacts the phenotype in the fish. And the reason to use the fish is that drugs can be tested very rapidly in the model. So it's a good first system in the whole animal context to test an idea. The other strategy, which is really where the power of the fish lives, is in what we call a non-biased drug screen, where we don't know necessarily what might help, but we're going to test hundreds or thousands of drugs to see if they can improve a aspect of the fish phenotype, by which I mean an abnormality that we would associate with muscle disease, like movement, or perhaps like the calcium imaging abnormality, something like that. And the advantage for fish there is that one can't really do these kind of large scale screens in any other vertebrate model in the intact animal. So you couldn't do a 1,000 drug screen in the mouse. It would just be time and cost prohibitive. So to talk about the targeted development, I'm going to present an example that is now quite old, but I think it's nice because it's made it all the way through to clinical trials, so you can see sort of the evolution of things. And this is using what's called the relatively relaxed zebrafish, which is a genetic model of RYR1 myopathy, where there's mutations in the zebrafish RYR1 gene causing its complete loss of expression. And these fish show characteristic abnormalities, including abnormal triad function, and have a reduced survival and abnormal muscle movement. So we did what's called a comparative array, where we took the normal fish and our RYR fish and said, what's different about them, using an older technology called a microarray. Still used in clinic, but this was a strategy that's not really used in the research basis as much anymore. What we found is that cellular stress was abnormal in the RYR1 fish. And in particular, we found that oxidative stress was increased. Oxidative stress is something that's been reported in other muscle diseases as being increased, and it's something that has a very clear number of drug targets. So there are several drugs known that reduce oxidative stress. So we tested one of those drugs in the following manner. We identified our RYR zebrafish, put them in this automated, or we put them in wells, added either an antioxidant drug or a placebo. So we did a placebo-controlled blinded study, and then recorded daily motor function for several days. And for this, we use our automated system. We used to use this Nolda system. We use a newer one now from a company called Viewpoint. It uses a laser to track fish movement. And for those of you that are Austin Powers fans, it's truly it's not a laser actually on the fish, but it is one monitoring the fish. And what we found, and I think you can see this here pretty clearly graphically, is that when we used an antioxidant called N-acetylcysteine, we increased the movement of the RYR zebrafish. So here's untreated, or the placebo treated, and 6 fish treated with N-acetylcysteine. So we could show, this is now looking at several hundred fish, that it was just a significant improvement back to normal movement in our mutant fish. And this gave us the confidence to think that this could be a strategy to move forward to patients. So to summarize, we found this abnormal pathway, oxidative stress, and we used a drug targeted at it to see if that would reduce oxidative stress, which it does, and improve the pathology in our fish. So of course, the question is whether this is relevant for patients. At the time that we did this, there was not a suitable mouse model for studying recessive RYR1 myopathy. Since then, we have generated one, and a couple other groups have as well. But at the time, we did not have that available. And so what we did is we looked at patient myotubes as a strategy for seeing whether oxidative stress was something that could be patient relevant. And we used the same assays we did in the zebrafish, and we found that in patient myotubes, there was also increased oxidative stress, and that we could prevent oxidative stress-related death in these patient cells using N-acetylcysteine. So when we put all this together, we felt like the oxidative stress increase was a conserved abnormality across not only zebrafish, but also human muscle, and that N-acetylcysteine seemed like a drug that could ameliorate it, and thus was a potential therapeutic for RYR1 myopathy. Based on this idea, a group in NIH led by Katie Mayer proceeded to do a clinical trial of N-acetylcysteine in patients with RYR1 related myopathy. I'm not going to go into all the details, but I will quickly summarize the findings. So there are 3 key findings. One is that much like in the patient myotubes and the zebrafish, patients with RYR1 related myopathies have increased oxidative stress, and it's something that one can measure in the blood. So it's clear that it's an ongoing process that seems to be important for disease. Number two is that unlike in the patient myotubes and in the zebrafish, the exposure to N-acetylcysteine in patients did not lower oxidative stress. So it seemed like the drug was not achieving the same changes that we would see in the patient myotubes. And then, as you might predict, because it wasn't actually reducing oxidative stress, we did not see, or there was not seen an improvement in motor function in the RYR1 patients treated with N-acetylcysteine. So there was a trend towards improvement. So if you look here at the 6-minute walk test, there was a non-significant increase of 23 meters with the drug, but it did not reach statistical significance. So the conclusion from this trial is that N-acetylcysteine was not successful in treating RYR1 related myopathy. And the plan at this point is not that oxidative stress isn't important, but rather this might not be the right antioxidant. So there's now new efforts trying to find a more potent antioxidant with better muscle exposure to use for treatment. And this is just the publication. And I think I should highlight this. Again, this was work that was sponsored, the original preclinical work with some contribution from MDA funding. And then this was an NIH-sponsored clinical trial. So quite a number of groups that contributed to this. So I lastly want to talk, I think I just have a few more minutes left, and want to talk about that idea of a large-scale screen. So this is really where the power of the zebrafish can be. And that's where we can test several hundred or thousands of compounds in a very quick amount of time, not knowing necessarily what might help, but using a disease-relevant phenotype as a way of screening. So in this case, we used our model of X-linked myotubular myopathy. And the screen that we did, we looked for drugs that would prevent the development of a phenotype of fin degeneration. So if you can look at the fish here, you see like this little sort of grayish outline. This is the developing fin of the zebrafish. And in the zebrafish mutants that model X-linked myotubular myopathy, this fin degenerates over time. And here you can see one that has basically no fin. And so our drug screen was based on this very robust and reproducible phenotype. And what we were looking for were any drugs that prevented this fin degeneration. And we did a large-scale screen of this Microsource US Drug Library. So actually, I think it's 1,400 compounds. These are all ones that have been in clinical trials. So they have shown safety data suggesting that they could be used in patients, again, without having to go through the preclinical process of all of the safety and toxicity studies. So this is what we call a repurposing library. And this is an example of drug repurposing. So our first screen gave us 38 hits. We retested them and found that 10 retested positive, where if we took 16 embryos, it prevented the fin degeneration in all of them. We've been focusing on one in particular, and because these is unpublished data, I'm going to just give it a generic name of SK01 for now. And we were able to show that SK01 in a dose-dependent manner could suppress the fin degeneration phenotype. So again, this is a validation of the drug screening result. And importantly, we could show that SK01 improves the motor phenotype. So this is a direct muscle-related phenotype. This is, again, using our automated fish movement assay. And we also see that SK01 increases survival in our XLMTM zebrafish model. So where have we gone from here with this? So we want to work towards clinical translation of this finding, but we're also using this as a springboard for better understanding of disease, because this drug is acting at a pathway that wasn't previously associated with XLMTM. It's providing us, we think, new insight into how the disease actually happens. So in terms of the clinical translation piece, what we've done is we've taken SK01 and taken it to the mouse model of XLMTM and done a randomized placebo-controlled trial where we give the MTM mice either SK01 or a placebo. And then we look at motor function and survival. And we did survival as our primary endpoint because the MTM mice only live about 35 days as compared to 2 years for a normal mouse. And what we found is that SK01 nearly doubles survival in the MTM1 knockout mice. And also improves motor function. This here is using a wire hang test, where basically we put the mouse on a wire grid and we turn the grid upside down, and normal mice will cling to that wire grid for several seconds, and often for even longer than a minute. And our MTM knockout mice can only do this, as you can see here, at 30 days, they only can do it for about 10 seconds. And we were able to find in a significant manner that the SK01 increased the wire hang quite significantly as compared to the untreated mice. So what are we doing from here with SK01? We are completing the pathologic and molecular analyses. As I mentioned, it's providing information about a new pathway involved in X-linked MTM, and that's a very active subject of research in the lab right now. We also tested another compound similar to SK01. We tested actually 8 of them in the fish, 8 different ones, and they all rescued the phenotype. And then a second one in mouse, and this one now we have reproducible results showing that it also doubles survival and improves the wire hang. The other thing we're doing is we're seeing if SK01 can help in models of congenital myopathy that are similar to X-linked MTM. And finally, we're thinking about whether or not this is something that we can consider as translation to patients, and we're in the process of trying to decide how that might go right now. I did want to highlight that we're not the only ones who have done large-scale drug screens in zebrafish, and this is not an exhaustive list, but just to know that this is a strategy that's being used by several different groups, and the findings have been really quite interesting, and have resulted in a couple drugs already being translated to clinic that started with the zebrafish drug screen. So to summarize, and I think I'm right on time, there are several different types of animal models available for studying human neuromuscular disease, and in particular for aiding in drug development. I want to emphasize that no single model is perfect, and that the key, and this is the onus really is on the scientists to do this, is to match the model with the question that you're asking. So for example, if the question is, how is this drug that I'd like to bring to clinic metabolized in a way similar to patients, so I can understand what its availability will be and what dosing I might need to use in a clinical trial, well, a fish isn't going to be very useful for that. A mouse might be somewhat useful, but a large animal model would be ideal for studying that, because the metabolism of drugs in dogs, for example, is very similar to humans. On the other hand, if I wanted to say, how can I find new drug targets, and I want to screen 100,000 drugs, again, that's not feasible in the mouse or dog model, but is perfectly amenable to a zebrafish model. And then obviously, depending on the question, the different models might be the mouse or patient cells. So just understanding that there isn't a one-size-fits-all. And I think more and more researchers are doing work as my lab does, which is using multiple models and just matching them with the specific question. And I do want to emphasize that while it is not perfect for all indications, zebrafish has emerged as a really outstanding model for studying neuromuscular disease. And the 3 uses where it's really been a shining model is with the validation of new genetic causes, elucidating novel pathomechanisms, and identifying and being the first step for identifying new potential therapeutics. I just want to thank my outstanding lab for all of their hard work. And I've presented just a few of the many projects that are ongoing. And I definitely want to thank all of the sponsors of our research, and in particular, Muscular Dystrophy Association, who, as I mentioned, I've been very privileged to have support from them for many years. And these are just a fuller list of the people that have done the work. And I should also mention that this represents collaborations across several groups at University of Toronto, but also around the world, including Bob Dirksen at University of Rochester, Alan Beggs at Boston Children's, and Karsten Bonneman at NIH. And with that, I will end and take any questions.
Thank you very much. That was so interesting. How many zebrafish do you have?
That's a great question. We probably have, so we do most of our studies on the embryos and the larvae. And so the embryos will be any fish up until day 4 after fertilization, and
Okay.
larvae are after day 5, day 5 to day 14. And we do all of our studies on those, but we house the adults because that's how we generate the embryos and larvae. And we probably have close to 20,000 adult zebrafish.
Holy
We
cow.
currently have zebrafish models of X-linked MTM, centronuclear myopathy, Nemelin myopathy, RYR1 myopathy, a couple forms of congenital muscular dystrophy, and of Duchenne muscular dystrophy.
That's amazing. That's amazing. So I'm just curious, from researching and following all of the zebrafish, uh, what's the time frame to go from monitoring and studying those fish to actually getting a clinical trial off the ground because of what you've learned?
Yeah, that's an excellent question. So, I think that, for us, the one example so far has been N-acetylcysteine. We published that data in 2012. We probably made the initial observation in 2010 or 2011, and the clinical trial was completed last year.
Okay.
And that's a drug that we didn't have to do any additional preclinical toxicity or safety studies. We used dosing that was already known in humans, so there was no additional development process from that perspective.
Mm-hmm.
On one hand, that accelerated the timeline. Things were slowed by a lot of challenges with recruitment and on the clinical trial end that were somewhat unforeseen, but I think have been encountered with many different trials. What is the fastest that something could be translated? So for example, for this new drug screen, we completed the drug screen including all the validation, within 6 months, and completed the mouse testing in another 6 months. So we're at the point now where we could start thinking about translation to patients, but then there'd be at least a year of preparing for a clinical trial, developing a protocol, applying to FDA for either an exemption or for an IND, and then actually starting a trial. So, I think realistically, if one had the most rapid development, you're still talking about probably 3 to 4 years from discovery to patient, which is, compared to what historic norms have been, is really fast. But I know for families and for patients, it also seems to be excruciatingly slow.
Yeah.
But there's a lot of important steps that are put in place in terms of validating findings, but also preparing for a safe clinical trial that are really critical.
That's so interesting. Thank you very much for presenting that. And thank you very much for your time this morning on a Saturday.